Hi, everyone. Thank you so much for joining us today. We're going to get started in a minute and a half, 2 minutes. We'll just give everyone time to hop on and get settled. But in the meantime, if you want to share in the chat your name and the university or organization that you're a part of, I think that would be great, or the location where you're joining from, whatever you would like. So awesome. Barstow College, our friends from the Lrnr team joining from the Bay Area and DC, Drake University, Midland in Texas. Awesome. No, I'm so happy that so many people are joining us from all over today. Oh, Denmark. Awesome. Richmond, Texas again, Puerto Rico. Wow, we really are coming from all over. Well, thank you all so much for joining us today from all these different time zones and locations. We really appreciate it. All right, since we've given everyone a few minutes to hop on, I think that it's good for us to go ahead and get started. So I'm going to go ahead and share my screen here. Perfect. So hi, everyone. My name is Lindsay, and I'm a marketing specialist at Openstax. And today we're joined by Arvind and Clifton from Lrnr, who will be talking about teaching biology labs using Lrnr virtual experiments, 2D and 3D models, adaptive quizzes, interactive lecture slides, and more. So a lot of you are probably familiar with Openstax, especially if you've come to Lrnr's other webinars that they've held this year. But if you're new to Openstax, there's a few key things you'll want to know. So Openstax is part of Rice University, which is based in the wonderful city of Houston, Texas. And we are a nonprofit organization. We're able to offer our library of free textbooks due to philanthropic support, such as from foundations like the Bill and Melinda Gates Foundation, individual donations, and mission support fees from our technology partners like Lrnr. So whenever an Openstax user pairs one of our free textbooks with one of our paid technology partners like Lrnr, the technology partners give us a small mission support fee back that allows us to sustain our operations. Openstax is mission-oriented, and our aim is to transform learning so that education works for every single student. Since 2012, we have published over 45 free, openly licensed, peer-reviewed textbooks, in the process saving students over $1.7 billion. Now, you just heard me say that Openstax publishes free, openly licensed textbooks. But what exactly does that mean? So Openstax textbooks are one example of OER, or open educational resources. And we have a wonderful definition up here on the screen from the William and Flora Hewlett Foundation, which is a super strong supporter of OER. But in essence, OER are learning materials that are available in the public domain, don't cost anything to use, and lack traditional copyright restrictions, so that users are able to edit, revise, remix, and redistribute openly licensed content however they'd like. Here at Openstax, like I mentioned before, we offer a library of over 45 textbooks in the sciences, math, social sciences, business, humanities, and college success. Each of our books meet the standard scope and sequence requirements of their respective courses and are written and peer-reviewed by industry experts and scholars. Each of our books undergoes a rigorous editorial process, and we make regular errata and industry updates when necessary. So for instance, instructors have the opportunities to submit suggestions for updates on our website. In addition, we publish new editions of our books and make revisions when it's pedagogically necessary. So for instance, if there's a major development in a field, we may publish a new version of one of our books entirely, like we recently did with our astronomy textbook. Meanwhile, if there's just smaller updates or revisions that need to be made, we make those in the online versions of each of our textbooks. Overall, we aim to revise our textbooks to make sure that the content is up to date, relevant, and diverse and representative of different audiences. Overall, OpenStax textbooks are a lot like other textbooks that are available out there on the market. However, unlike textbooks that aren't OER, OpenStax textbooks are free and are openly licensed, that you can edit and revise the content however you'd like and in any way that best meets your classroom needs. We offer a lot more than just textbooks though. So each of our textbooks comes with free instructor and student resources like LMS course cartridges and course shells, PowerPoint slides, and test banks. Additionally, we offer free webinars and free articles on the OpenStax blog about a variety of different topics, such as the best technologies to pair with OpenStax products, tips for teaching, and tips for using our products. We also offer OpenStax Tutor for some of our books, which is a courseware platform, and we also offer our support center. So our support center is an excellent resource to turn to if you have any questions about our resources or just want to learn more about how OpenStax books are created. Lastly, we offer OpenStax Tech Scout. Here at OpenStax, we understand just how critical education technology tools are to learning nowadays. And we also know just how challenging it can be to find the right education technology products for your course. Therefore, we created OpenStax Tutor to help alleviate this. OpenStax Tech Scout to help alleviate this. And OpenStax Tech Scout is a searchable tool on our website where you can filter for different courseware, lab, online homework, products, simulations, etc., that both meet your classroom needs and align with our different OpenStax textbooks. And Lrnr is one of the amazing learning technology companies that we partner with and that you can find on Tech Scout. So Lrnr offers low-cost, high-value options that ultimately support instructors and students. And Lrnr helps to enrich the OpenStax experience and helps to enrich OpenStax materials and products through the financial support that they're able to give us. And in effect, the support that Lrnr gives us allows for OpenStax to keep our books current and accessible for all. So now that we've dived a little bit into what OpenStax is and our partnership, I'm going to pass it off to the Lrnr team.
Awesome. Thank you so much, Lindsay. Let me just share my screen real quick. And let me know if you can see this. I'm just gonna do a slideshow. So, hello everyone. Thanks so much for joining us. My name is Arvind. I'm from Lrnr, as Lindsay pointed out. We're here to talk about how to teach A&P biology labs in conjunction with the OpenStax, whether it's the Biology 2E textbook, the concepts of biology, or the microbiome. So we are actually building these virtual labs that align to the OpenStax text. So we'll just go through some quick facts about Lrnr, talk about some best practices that a lot of the instructors that are using OpenStax and Lrnr have been employing in their class and also in the labs, followed by a brief demo, and then a summary, and then some Q&A. So we are a social impact startup. Our focus is to democratize the cost of course solutions without compromising quality. My background is predominantly from Pearson. I built a lot of the platforms that you know, like Mastering Biology, Mastering A&P, MyMathLab. So I was at Pearson for over a decade. I was the chief architect for the last 3 years before I left Pearson. And this was almost a decade ago. And obviously, all these platforms have evolved over time. My biggest challenge was to convince the top folks at Pearson to basically make content as cheap as possible and focus on student success. That is through the homework and classroom engagement tools that we were building out there. Anyway, so in 2016, we partnered with OpenStax, and we had built a platform which needed content, and OpenStax provided us that the content that's needed. And our focus was purely to create a high quality formative assessment and provide a complete personalized course that included homework solutions, classroom engagement tools, and also make sure that all of this addresses the course and sequence needs for your individual courses. So our goal for the next 3 years is to be able to provide you with all 100 to 300 level courses for more like a Netflix model, $80 for the year for all courses. So you don't have to worry about, students don't have to worry about, if I take this course, what's the cost of the textbook and what's the cost of the course material, the homework solution, and so on. So every single course we build is literally a custom course that aligns to your pedagogy. So we can literally take all the our collection of courses that we have, even though it aligns 100% to open ed, and 95% of the content is actually OpenStax when it comes to the learning content. We can mix and match. We can literally take chapters and sections from any titles and make that into your course. Our focus, like I said, is formative assessment or formative content. So all the questions and activities that we've done are from the ground up. We do not use any of the OpenStax banks. Our focus is to make sure that students can start thinking critically. So 70% of the questions that we've written are written at a higher level of Bloom's. All of our courses you can integrate, they integrate very well with most of the major learning management systems like Canvas, D2L, Moodle, and Blackboard. To make sure that all students have access first day of class, we do have a 2-week trial to ensure that all students, obviously who might be waiting for their financial aid and, but they still need access. We don't want them to lose out. We have a customer support 7 days a week during the first 6 weeks of the semester. That's typically when most of the students try to join. And if there's any issues, registration issues, trying to do their first homework, we'll try to resolve that. And then we are available 6 days a week for the remainder of the semester, typically on a Sunday for the weekend, because most students come on a Sunday last minute sometimes to finish their assignments. And finally, we do have a learner give back program because we work predominantly with community colleges and in a lot of areas where there's students that are disadvantaged. We have this program where instructors or departments can tell us that these are the students that actually need free access. And we use that, whatever revenue we've generated, we take 5% of that, earmark it, and say, okay, we will give free access to those students. So some of the stats, so the virtual labs or the biology labs started as a grant project. It started as a A&P grant with the Department of Minnesota Education. So we won a Shark Tank Award, and then we won a grant from them. And then later on, we also, with CUNY, we also got a grant from the CUNY system. So we're working with CUNY hostos, along with Truckee Meadows Community College in Nevada and Pima, to build virtual labs. We've already built 23 virtual lab experiments. There's 20 more, and we'll go into the details of that. And along with that, that aligns to the biology majors, biology 2E, also the non-majors, and also some microbiology topics, if you will. These are the stats for our various courses, if you will. We also have an environmental biology course which is also a mix and match of OpenStax and other OpenEd resources. And sometimes we actually build our own content, even though we are not a true traditional publisher who builds textbooks. But wherever we find gaps, we try to fill those gaps. And if there's certain topics missing, we'll add those topics. All the labs, because of the grants have subsidized the cost of development, all the labs are $20. It's not per lab, it's for all the lab experiments, the entire course. The lab course is $20 for the semester or for the term. And if the student flunks, basically, or drops out or gets an incomplete, they have up to 12 months to retake the same lab course without having to pay us anything extra. All of our lecture courses, premium courses, are $40 a piece. It has a lot more content, and I'll show that to you. And then we have our library model, like I said before. So all the courses that you can see here, they're all at $80 for the year for all courses. So essentially, this works out really well if your department or multiple departments start using Lrnr. And for that $80 per seat, students don't have to again think about, for anatomy, what do I pay? For biology, what do I pay? For math, what do I pay? And so on. And like I mentioned before, the biology labs course that we have is tightly aligned to the OpenStax textbook. We have an adaptive sidebar. So what we really do is we take the textbook, we curate content like videos, terms and definitions. We curate such content, and along with that, we create our own activities, whether it's in 2D or 3D or quizzes, and we align that to specific learning objectives and specific topics in the textbook. So the adaptive sidebar adapts to what the students are studying in the textbook. Just like every other online e-reader, you can annotate, highlight, bookmark, take notes. But apart from that, we also, in the process of a student is building, basically creating their highlights, notes, and bookmarks, we build a study guide for them. The study guide is an aggregation of all the highlights, bookmarks, and notes neatly organized by chapter and section. So it becomes easy for them to, for their midterm or final exams, they can refer to all their notes rather than going into the textbook and then trying to figure out where the highlights are. We have already organized it in a study guide, and then they can refer back to the textbook from their built study guide. So I already mentioned before about the focus of building formative assessment for us. And we also have multiple question banks. One of the banks is hidden. So unless you actually use it in a homework setting, it's not going to show up for students for practice. And 99% of our questions have wrong and right answer feedback. And most of our questions are linked back to specific areas in the text. So essentially, it's more like, it's a remedial help. So not only do we give wrong and right answer feedback, which is a first level of remediation, but then if the student still doesn't get it, there's a little link that'll link them back to the text, and I can show that to you as well. You can build assignments on Lrnr. As you know, I'm preaching to the choir. Without building assignments that are gradable, nobody's going to do it. It's very easy on Lrnr to build assignments. In fact, if you're already familiar with the OpenStax textbook, you don't really need to explore Lrnr a lot. You can simply start building an assignment, and in that process, we will preview. You can preview pretty much all the activities, virtual labs, videos, everything that we have that aligns to the topic. You can also build templates and reuse those templates across, multiple semesters, and you can also share them with your other colleagues, so they don't have to duplicate effort. And if you're using Canvas, we also build deep links. That means that we create a link for every single assignment you build, and that can go and sit, the hot links can go and sit in your Canvas course. So essentially it becomes very easy for you to manage your assignments right from your Canvas course. So in the last 1 year, we started to also focus on classroom engagement. Actually, it's more than 1 year. It's been 2 years in the making, but we just released it a year ago. Because of COVID, everything started going online, and a lot of instructors started opening up Zoom conversations like this, but they wanted everybody on video while they were obviously giving a lecture. And it was very hard to manage such a class and engage students because you have no idea whether the student is actually listening to you or just doing their own thing. So live polling and lecture slides, where you can upload your lecture slides, embed activities in between the lecture slides. Very easy to build those kind of activities on Lrnr. We thought that rather than you guys going to Zoom, using the Zoom live poll or Kahoot or Top Hat or Nearpod, again, another point solution that the students have to learn, we said, why don't we build everything as part of the same experience? And that's what we've done. Again, the goal, so let's talk about some of the best practices in a minute about both live polling and lecture slides. All the content that we have built is HTML5 based. So except all the 3D models and image identification based activities, most of the content on Lrnr is ADA compliant. The 3D model based activities, as well as the identification of an image based activities, if you define or explain the image and the activity is more about identification, it defies the purpose of the activity, and hence it's not ADA compliant. So best practices that a lot of our instructors have been using for both virtual labs, histology, and 3D models. So virtual labs, the way that we started focusing on virtual labs, of course, a lot of the design of these labs was in close coordination with lab coordinators across multiple systems. So the main goal for us was to reinforce key concepts. I'm a strong believer that you can't, learn swimming unless you fall in the water, right? So here it's more about following the procedure, because most students do not follow procedure and hence don't get the right results or the expected results, and they start asking why, because they're not paying attention. So what we've done is, rather than trying to build an amazing virtual, or augmented reality or virtual reality based experience, everything that we've done is very 2D-ish. So it can touch, folks who are 50 years old or 18 years old, who have not played any of these games before. And the goal is to deliberately focus on the procedure. They don't lose any points for for basically following the procedure. If they don't do it right, they get to do it again. So the experiment will not move forward without that. I'll show that in a minute. And then engage the learners more on concepts. So rather than showing them what happens, we'll ask them to hypothesize on what is going to happen. Let's say in the Benedict's and the Buret's test, we'll ask them what color does the solution change to after giving them scenarios, and the test tubes and the test tube values are not the same across multiple students, and that's why it's not static. So you can't just take screenshots of it and then give it to another colleague and they can just copy the values. So and finally, to reduce a lot of waste in the labs, quite honestly. So for A&P labs, we do have histological slides where you can zoom in up to 40x. We're also trying to get some plant tissue based histological slides, and we're in the process of doing that. But for now, we have a lot of activities that are aligned to human histology, and I know that there is a chapter on animal histology in biology. So again, this might be useful for you in the labs. Again, I'll show it to you and see if this can benefit any of your students. Basically, it's to bridge the gap between structure and function. And finally, 3D models. I know that in biology, of course, we're talking pure biology, but there are some chapters on anatomy. It's not only human anatomy. I think it's about all anatomy, but we do have models for the human anatomy. So it's something that you might be, able to find it useful for your students. Again, it's there if you want to use it. So time for a demo. Let me just switch gears real quick. Okay. So each course, so this is our courses page. And then from the courses page, I'm just going to Biology Labs. And imagine this is the OpenStax textbook that's enhanced. So when you click on the textbook, you do get the table of contents, the way we organized it for labs. But then remember, I told you there's an adaptive sidebar. So that adapts to whatever, as I'm scrolling through content, content keeps changing on the right. So it's aligning, it's very contextualized to whatever I'm doing here. Okay. So let's actually go to there's multiple chapters and sections here. I'm on the cardiovascular system, which is not really relevant to you guys. So something like that. So on the right-hand side, you see terms and definitions. There's a quiz. There's a drag and drop activity, and so on and so forth. So let's go to one of these activities. Let me actually take this out of the way because I cannot do this without this is there in the way. Okay, sorry about that. So this is a drag and drop activity. We have over 10 different activity types in the 2D space. And in this case, I'm just dragging and dropping stuff onto an image. So these are simple activities, right? So we built a whole bunch of such activities. We also take care of students who are colorblind. That's the reason why there's also a strike through. And students can do all these activities by themselves. And if they don't know some of these activities, they can simply hover on the label to see how they can do these activities. And Lrnr is really good with help, right? So you can click on this help button and it takes you to a contextual page that shows you how to do that type of activity. So it doesn't matter if you're an instructor or student, whatever you're doing, the help button is contextualized to the page that you're on. Okay. So from here, I can go back to the text. And let's go to a different chapter. There's quite a lot of drag and drop activities in there. Okay, and now finally, there's adaptive quizzes that I'd like to show you real quick. Let me go to one of these and then, sorry, this is again A&P text. So in CUNY, their biology labs comprises of both A&P and biology. And me not being a biology major of any sorts, I'm trying to find something that's more relevant to biology and not A&P. Okay, so there you go, plant tissues. It's probably easy. Okay, and I'm trying to find if I can find, these are all activities that students can do. And the easiest way for me to go to some of the quizzes that we have. There you go. So that's a hybrid question that I just showed you. It converted to a multiple choice. So if I don't get it right, I literally can click on this reference link, and it takes me to a specific area in the text. And then after I do that, I can always get back to the question, answer the question, and then move on. So essentially, every single question has, most of the questions I would say have right and wrong answer feedback, along with the link to the text where students can go read the text and then come back and answer the question. Okay. So we also have a whole bunch of activities, and then finally we'll go to the virtual labs and simulations in a minute. So again, there's a ton of questions and activities that you can assign. You can consider this as more of pre-lab activities and quizzes. And then finally, let me show you all the simulations that we have. So this is going to be a combination of both biology and a bit of A&P. And we do have 23 of these simulations. In fact, we just completed one more. We updated our streak plate method with a new one. Again, a lot of these don't make sense to me, but I just learned the streak plate method, so I can hopefully give you a good demo on this. So let's actually resume the streak plate method. So it actually starts with start with the introduction. So every single experiment that we have broken down is introduction, preparation, procedure, and observations, where we ask students to hypothesize. So I think I was told by our content subject matter experts that students make a lot of mistakes in streaking, right? They don't basically, they don't look at they don't basically sterilize, make the inoculation tube, or whatever you want to call it. They don't sterilize it every single step of the way before creating a streak, and that could cause a lot of problems. So we show them the steps with a video, and then the students can actually see what are the different items needed, and then they can go to the procedure. So in here, use the inoculation loop, put it on the Bunsen burner, then turn it off. Then again, you can't just put this immediately into the stock culture. Okay, it's going to kill the bacteria, I guess. So you need to wait, and once it cools down, then put it in. So evaluate that. So again, it's a lot of following the process. So in this case, it's a continuous streak that I need to use. Thank God, last time I was doing some other radiant streaking or something, and I was getting it wrong. So in this case, it's super simple, thankfully. And then once I do that, so I was told again that after you do all the streaking, you're still supposed to sterilize the loop and then keep it somewhere, I guess. So that's what I had to do. If it was a different type of streaking, I had to do the streaking between every streak. I have to do the same process, and then again sterilize this and keep it aside. So now cover the Petri dish for incubation. So let's go to observations. So after the incubation, which area of the streak will have the highest bacterial growth? I would say hotspot 3, I guess. Okay. Now I don't need to get any more right because I got 1 right. That's good enough. Submit answer. So again, you get a whole series of questions after you've done the procedure, and this is where you get points. So the points are based on the procedure, and then the first question typically always varies. This is for the streak plate method, but in the other other experiments, it's a little different. So let's actually go through some of the other experiments as well. Okay, I'm going to exit this and I'm going to show you a different experiment. So photosynthesis is probably easy for me to explain. I thought I'll do Hardy-Weinberg, but then I got confused. So I'm not going to even try. So there's a carbon dioxide source. So again, it's all about telling the student what they need to prepare. So initially it would start here. And you start the experiment, you go through that, and then you continue with the procedure. Again, in this case, it's a whole bunch of instructions. And then we'll add the lights. And I've been told that students don't even match the lights sometimes when they're doing these experiments. Just kind of don't understand why, but anyway. So blue is to blue. And let's say I put green here. I try to validate, it'll error out. And then I just have to change it, validate it. So, so this is where I talked about hypothesizing, right? Rather than showing the students as to, okay, for which one of these incubation thing, where will it float more than the rest? We'll ask the students to hypothesize, ask them to use a little bit of their brains, and hope that they get this right. And I'm going to randomly pick stuff, submit my answer. And then the next question is based on now we'll show them the correct answers. And then we'll ask them to hypothesize where is it present, where is it absent, and so on. So there you go. That's how, that's my first question. And the second question is reasoning behind it. This is what I would do if I'm a backbencher. There you go. So I got 60 out of 150. And all of these lab experiments are assignable, right? So that's one of the nice things about this is when you assign these lab experiments, for some of the experiments, students won't get, if it's test tube based experiments, they won't get the same test tubes with the same scenarios or the same values. So they just cannot copy from each other. And there are going to be others where they can. We also have something like microscopy. Where you can pick the different slides, I guess. I guess next. And then slide chosen. Turn it on, specimen centered. Okay. and look through the 4x lens. So here you can adjust the oculars, I guess. To the course focus. Ah, something happened. Then there's an adjust iris diaphragm. What is that? Okay, that's this. So everything here has to kind of check out. And then I have to do the same thing for two others. I'm not going to waste your time or my time in doing this. And then, and finally, there's a question at the very end, which is 10 points, which is to calculate the magnification. So again, the goal of this microscopy experiment is to just get familiarized with the microscope and all the different functions of the microscope and how you do it. So that's the virtual labs. And of course, if anybody has an interest in looking at more, just ping us and we'll give you access and you can play around with it. And then finally, when it comes to live polling, before I go to live polling, let me just show you how assignments are built. So you could literally create an assignment. You could either create a homework or a test on Lrnr. So let's say you don't want to use responders or honor lock. And again, trying to build your own tests, copying all the questions, it's a lot of work. So with Lrnr, you don't have to. You can actually use our test mode. And in our test mode, if you have multiple windows like this open and if a student has started the test, some students, they're, pretty smart, right? They'll try to open up multiple windows of all the homework and everything else. And they'll say, okay, I have all this homework open now. Let me just do the test. They'll be surprised that every single window will then go back to the test. So when they start the test. So we can give a name to this. Here you can give a whole bunch of instructions, links, whatever you want. And like I told you before, this is going to be the custom table of contents based on whatever course we've built for you. So in this case, I think I go to plant tissues. You can literally tell your students what to study and not study. And then even with videos, you can preview all the videos that we have curated in one place. Just out of curiosity, I'll just show you how that works. So you preview it here. You can watch the video. All of our videos are ADA compliant. They have closed captioning and the whole 9 yards. Again, it's not videos we've created. It's basically things that we've curated based on what instructors have told us. It could be from Jive, it could be from BC Campus. It could be from multiple YouTube channels where we've actually pinged the authors and said, hey, is it okay to use these videos? Activities, the activities that I just showed you, you could literally pick any of those activities and then select those activities and then use that in your assignments. So remember I told you there was a hidden bank of questions to maintain academic integrity. So we do have a homework bank or a homework bank quiz from which you can actually pick your questions more for a test maybe. Right? And then finally, the lab experiments, the ones that we just did, you can assign those as well. I don't know what's relevant to seed plants here, but just going to go with mitosis or something. And then finally, the step 3 is to review what you've told them to study. Activities, if you don't grade them, apart from the top 15 or 20% of the class, I think most of them will not do it. Number of attempts, You can give them some grade points. You can even select or customize the activities. You could build your own activities on top of this. So when you expand these, you could see that you could pretty much preview all the activities. You can literally click on show answer because we're not trying to test you. We want you to be able to quickly select these activities and questions. So if you feel that some of these questions or activities are relevant, you can use these for your custom quiz. If you want the students to do all of them, you don't have to do any of this. You can just come out of it. So let's just save this for now. And it says customized at the very end. Practice quiz is the same thing. You can, by default, this is an adaptive quiz. Typically, students get only 6 to 10 questions. You could say, you know what, I want to pick my own questions. So let me zoom in a bit. So the questions here are organized by Bloom's level. So the highest level of Bloom's comes to the top. So you don't have to really search for questions. All the memory-based questions go to the very bottom. And this is where I told you that you can build your own questions. So that's part of my bank. So let's just look at the very top. You can click on any of these questions. You can see the distractors. You can see the feedback. And if you don't like the question, or if you think there's an error with the question, you can click on this little bug icon and report an issue. It could be inaccurate, there could be a typo. And within 48 hours, we not only fix the issue, we basically fix the issue and we'll notify you. And if there's any students who've already taken this particular question or activity, we'll also give them the points so you don't have to do a lot of work. Like I said, our customer support is really good. So anyway, you can pick some of these questions once you've reviewed these questions, or if you leave it, it'll stay adaptive. Save. So that's how you can, custom pick the questions that you want to provide. And then the lab experiments, you just selected mitosis, how many attempts you want to give the student. So mitosis, again, it's one of those experiments where it's not static. So students get different slides. So they'll be kind of surprised they cannot copy. And then finally, let's give us some points here. And step 4 is to select. So let's say you're teaching more than one class section. You can select all of that and have different release dates. The start date can be in the future. And that's the date that the student actually sees the assignment. And the due date again can be sometime in the future and so on. You can also extend it for a specific student. And again, I'll not go into the details of this in the interest of time, but you get the idea. If you are interested, you could always talk to us and we can set up a detailed demo for you guys. And finally, I'd like to show you live polling a bit. So I'm going into Majors Biology because we've already built some slides. It's called Live. So you could literally build your own quizzes and you can upload your slide deck. And we have multiple activity types that you can build, everything from short answer to diagram-based matching, dropping a pin, and we are expanding this platform a lot as we speak. And the nice thing about live polling is you can actually provide participation points. So students, you don't have to penalize them. If they don't get it right, then you don't get any points. If you just give them some participation points, that's a way to give some carrot to make them attend, pay attention, and so on. And you can beam this just like any other live polling solution, and then students can pick up their mobile phones, go to the assignments module in Lrnr, and then join the poll. Slides work very similarly. This is a Openstax slide deck. So we just uploaded it. And in between the slide deck, you can embed an activity like this. So this is a diagram-based activity. And you can either make it into a matching activity, a clicking on target, a fill in the blank, drag and drop, and so on. So every single activity has a different way to rendition itself. So it's up to you. You're kind of the queens and kings of your kingdom. You can do whatever you want. Okay. So in the interest of time, if you need a demo on live polling, please feel free to connect with us or join us on one of our future webinars where we'll talk a little more about live polling. Let me get back to our assignments real quick to show a little more, and then we'll go to Q&A. So once students are, you published your assignment, they've done the assignment, you get a whole bunch of reports. And let me find, there you go. So the chemical foundation of life, this is actually a test. This is photosynthesis. Let's actually look at the report here. So your assignments, they can have multiple activities, virtual labs, quizzes, all of that combined into one single assignment, and there can be a grade point. And in this case, Hannah, you can look at every single student, and if a specific student comes to your office during office hours, you can literally review this with her and see where she's struggling, and then help her out after you look at the responses that she's provided. And again, that's to do with any student. I just picked this first student. Okay, so having said that, let's get back to our slideshow. So these are the labs that are currently in the works. So these are 20 more labs that this is a 3-year grant program. We are currently in the 4th semester of the grant program. We have 2 more semesters to go. And we are going to, we were supposed to only build 10 of these, but we found that all these 20 made sense. So we are trying to build all these 20 and add that to our list of virtual lab experiments. And again, the price is going to remain constant. It's not going to change. So in summary, we can build custom courses for you, and then you can assign your quizzes, activities, and virtual labs. You can conduct live polls with or without slides. We can integrate with your LMS where there's single sign-on, seamless single sign-on, and gradebook sync. So the grades automatically sync to your LMS. Student gets 2-week trial, and then we do have excellent customer support, and we do have a learner give back program. You could go on the OpenStax Tech Scout and see all the other instructors that have been using us and their reviews and what they're using it for, to get a sense of our quality of our product as well as our customer support. And if you would like to use Lrnr on a trial basis for summer, because you've attended this webinar, it's free of cost for your students for the summer semester. And please schedule a demo with us if you need to know more about the free pilot. We'll provide you a lot more details, and then if you want to test drive Lrnr, we'll give you a demo account so that you can play around with it. And after that, we can work with your admin, the LMS admin, to do the integration and so on. And we'll also onboard every single instructor that you have. So if you would like to talk to us, please connect with us. You can either schedule a demo directly on lrnr.us/demo, or you could send us a note. And I'm open for some Q&A. And so let's see. Okay, so I want to kind of focus on this. Interesting. By the way, please don't curse me. I am not your biology expert. So, so Deborah, I'm not your perfect biology student. I'm more of a physics and math student. So I tried to do my best. My apologies if that was not good. And we'll also update the simulation based on the feedback that you provided. Oh, okay, yeah. I mean, I think that also is something that we can do. So if you want to, based on the magnification, if you just want it to be different lengths, just to give a visual indicator, that's also great. Okay, so again, anything to do with microscopes, please provide us any feedback you have. Please connect with us. And if you have feedback, we'd love to take that because we are still in the mode of development. So essentially, we can always update. In the last 4 semesters, we have been getting a lot of really good feedback, critical, actionable feedback, right? If you just criticize us and say, this sucks, we don't know what to do. But if you criticize and then tell us exactly what can be done to fix it, then we're all ears. We'd love to, we'd love to get that kind of feedback. We do not have labs for environmental biology. Just looking at the questions here. Yes, so you can add your own questions. So you can create your own questions, take our questions, do mix and match, and all of that. Are there any other questions that that you folks might have? I'd be more than happy to answer those questions. Or again, please talk to us and schedule a demo with us, or just set up a 15-20 minute meeting with us, and we can talk further and also address any of your questions. Let me stop the share so I can see more questions. You cannot edit our question because we calibrate our questions for adaptive quizzes. That doesn't mean you could create a question. In fact, for live polling, so I'll take that back. As part of the next build for live polling, we are building a capability for you to import questions that Lrnr has, and then once you import the question into live polling, then you can edit that question. But you cannot edit the question that's part of the assignment. You can always build your own questions and then use that. So if you find an error, I did show you again, there's a little bug icon against every single question. You click on that, and within 48 hours, you just tell us, okay, this is the issue, and this is how you fix it. It goes to our editorial team. Again, I don't do the fixing because I suck at biology. So it goes to our editorial team. They're subject matter experts in biology, and they'll tell us what to do. The content engineering team will fix it, and they'll notify you. Yeah. So it takes place in 48 hours. All right. So thank you so much. And again, please send a note to learnfast.com, adopt@learnfast.com, or you could literally book a demo at this URL, and then we can take it from there. If you have any additional questions, I'd love to answer those.
Thank you all so much for joining us today. And thank you, Arvind and the rest of the Lrnr team, for yet another fantastic webinar. I always learn something new and exciting and innovative. So thank you so much again. We'll be sending out the recording of this webinar tomorrow, so you'll be able to watch it whenever you need and share it with your colleagues.
Hey, thank you so much, Lindsay, and thanks to the rest of the Lrnr team. Till we meet again.
Of course. All right.
Bye.
See ya.